Okay, we'll go ahead and get started. Welcome, everyone. I think I know or have met all of you. My name is Jen Schaefer. I teach the neurobiology course here. And it is my pleasure to welcome Dr. Sarah Heilbronner to give us a seminar on her work this morning. Dr. Heilbronner is an assistant professor of neuroscience at the University of Minnesota. She's also a preceptor for the MD PhD program with the U of M Med School. She earned her bachelor's degree in psychology from Harvard University, then went on to earn her PhD in neurobiology from Duke, and then completed a postdoc in the lab of Suzanne Haber at the University of Rochester in New York. Dr. Heilbronner has a wide array of publications in both sort of psychiatry type journals and neurobiology, kind of neuroscience oriented journals. The list is longer than I'm going to read here, but these are journals like the Journal of Neuroscience, Behavioral Psychiatry, Nature Neuroscience, Science Advances, those kinds of things. And I'm really excited to have Dr. Heilbronner speak. I had invited her to come give a seminar last spring when she was going to come up and see our beautiful campuses, and then COVID hit, so of course that didn't happen. So now we're trying again, and you know, it's via Zoom, but we're still happy to have her here. I'm really excited because her research, I think, bridges a really important gap in the field of neuroscience, where she's really trying to look at how we translate cellular circuit level data from model organisms into human and apply them to human psychiatric conditions and diseases. Specifically, she looks at the neural circuits that underlie motivation and decision-making, which have been implicated in a number of psychiatric disorders. So this is very timely research, and I'm very excited to hear about it. So with that, I think I will turn it over to Dr. Heilbronner. Charlotte, would you mind turning off both sets of lights in the classroom? And you can go ahead and, I think, share your screen. I guess I should have asked, do you have any sound that you need to share?
No.
Okay, all right, sounds good.
Okay, thank you so much for that introduction. I'm still sorry that I don't get to come up and see your campus, but maybe someday. And I'm really looking forward to chatting with some of you after this. Like Dr. Schaefer said, I did my undergraduate degree in psychology, and then a PhD in neurobiology. And now I feel like I work kind of at the intersection of those and more applied fields like psychiatry. And so if you have questions about how to pursue each of those different avenues and what they look like, I might be somebody that maybe those are conversations we could have in the following hour. All right, so I am particularly interested in the wiring diagram of the brain and what that means and what it can tell us about the brain's function and health and disease. And so first I want to tell you like what I mean when I say wiring diagram. So you guys all know that cells in the brain are special. Neurons are optimized to transmit information. They talk to each other. And so neurons sort of have the equivalent of ears, which are dendrites, and they have the equivalent of mouths, which are axons, right? And they fire action potentials and they communicate information back and forth. But the important part is to remember is that not all neurons in the brain are capable of talking to all other neurons in the brain, right? And you guys probably realized this as you went through your studies, but never really said it explicitly out loud. Not all axons connect to all dendrites. And so that pattern of which neurons talk to which other neurons, I'm going to call anatomical connectivity. We call anatomical connectivity. And that's sort of the main measure, the main tool that I'm going to use here. And I'm going to try to convince you that learning about structure in the brain tells us a lot about function in the brain. So just to let you to sort of give you background about how we study anatomical connectivity before launching into what its applications might be, in order to look at which neurons in the brain talk to other neurons in the brain. Here's what we have to do. We have to open up the skull and inject a dye in the brain. And there are sort of two different kinds of these dyes. Some are taken up by cell bodies and then transported anterogradely along the axon. And so they they will show us which cells, where cells are projecting to, and others will be taken up by the axons themselves, and the dye will be transported retrogradely. And so they'll tell us which regions are giving input to the area that we injected. And so in order to visualize these dyes, we have to extract the brain and all of that. And so obviously, and this is an important message, this is something that just cannot be done in humans, obviously. This is something that we have to work with non-human animal species on, which means, and this is a sort of shocking thing to realize, with all of our advancements in non-invasive brain imaging technology for humans, we don't know the wiring diagram of the human brain. I'm going to say that again because I think it's important. We don't know, and right now we can't know the wiring diagram of the human brain, which is sort of a shocking realization because of all the fancy things that we think we can investigate about the human brain and do investigate about the human brain. But this sort of basic structural property is out of our grasp right now. Okay. So I'm going to tell you a story about when I was a postdoc and when anatomical connectivity became a tool that we could leverage to answer some questions and to model some systems. So I was a postdoc at University of Rochester, and we had a visiting postdoc from the University of Puerto Rico, and he came, and the labs were collaborating, and he wanted to use optogenetics, which hopefully you've heard of, but is a sort of circuit mechanism that is invasive, and you can do in mice and rats to selectively manipulate certain pathways in the brain. He wanted to use that to try to make a really good mouse model of post-traumatic stress disorder, and to then sort of try to cure it, right? And this was based on neuroimaging literature. So there was a finding from the neuroimaging literature showing that an area of the frontal cortex called the dorsal anterior cingulate cortex had reduced activation in post-traumatic stress disorder. And he just wanted to take a rat and reduce its activity in post-traumatic stress disorder, which is actually a relatively straightforward thing to do, using optogenetics or using any number of more traditional techniques as well. The problem was, he came to me and he said, "Oh, you're an anatomist, you'll know this. Where is the dorsal anterior cingulate cortex in the rat?" And I was like, I think I know, but I'm not totally sure. Let's go to the literature and find out. And at this point, not all of the journals were even scanned into the older articles weren't scanned into the internet yet. So we went physically to the library. I think that's the last time I had to physically go to the library to find journal articles. But we went there and we dug around. And what we learned is that it's actually a really, really tricky problem. If you wanted to know which part of the rodent brain is most similar to this part of the human brain, where would it be? It's very, very difficult. And part of that problem comes from sort of the basic shapes that are in the rodent brain. So you guys have probably seen pictures of human brains, and even in this picture you can sort of see, although it's pixelated, that human brains are very, very wrinkly, right? They have lots of gyri and sulci on the outside of their cerebral cortices. Rodent brains are not. They don't have those wrinkles. They just kind of look like misshapen balloons. And so the question is, how do you just kind of have this lump of tissue without any wrinkles to guide it? How do you figure out which pieces match with which other pieces? And again, it was so important because we wanted to model and then correct this particular part of the circuit in order to try to advance neuromodulatory treatments for post-traumatic stress disorder. All right, so why rodents in particular? So if rodents just kind of have balloon brains, then why even bother with them? Why not come up with a better species or whatever it is? Or why not just abandon them and work, straightforwardly in humans. Well, rodent studies, and probably you've read some of these papers over the course of the semester, or at least heard about them, sort of provide an important set of tools for behavioral, genetic, electrophysiological, and molecular tools that are not available in primates and definitely not available in humans. I mentioned optogenetics, there's chemogenetics, there's calcium imaging, there's just all kinds of, there's transgenic mice, right? There's all kinds of things that we can, all kinds of tools that we can apply in rodent studies that we can't apply in studies of humans or even in non-human primates. And so the problem becomes, how do you figure out which part of the rodent neural circuits are equivalent to which parts of the human circuits, to which parts of the primate circuits? Remember, humans are just primates. And that's sort of a fundamental problem for translational research. And by translational research, I mean kind of the pipeline by which basic neuroscience from, say, rodent studies becomes applicable to, to treatment of psychiatric populations, or even something in between, like even just to our understanding of the human brain in its healthy state. And so that kind of bridging of basic and clinical science, of different species and of different methods, all of those are sort of translational problems. And so this is an important translational problem. If only rats just sort of had a human face and they could just tell us which part, that maybe that would be easier, but that's not the case. And so the part of the brain that we were interested in was the dorsal anterior cingulate, which I mentioned as part of this post-traumatic stress disorder problem, belongs to a much larger brain region called the prefrontal cortex. Again, you've probably heard about it, but the prefrontal cortex is just all the stuff at the front of your brain, and up until you get to the motor areas, which exist sort of in the middle here. So the prefrontal cortex, which you'll see here abbreviated as PFC, it's a particularly tricky part of the brain in order for figuring out whether the rodent has those pieces or doesn't have those pieces. Like I'll sort of contrast it with, say, the brainstem. There are parts of the brainstem, like the dorsal raphe nucleus. I'm just naming a random part of the brainstem. We're like pretty sure where that is in humans. We're pretty sure where that is in monkeys. We're pretty sure where that is in rodents. We're actually pretty sure where that is in almost every species that we've studied. And so that's not the major challenge. The major challenge comes when you talk about the stuff on the outside of the head, that wrinkly stuff, the cerebral cortex. And it's even harder when you talk about the prefrontal cortex. We generally think about the prefrontal cortex in humans as being important for emotion, for higher cognitive function, for memory, for learning, for decision making. If you remember back to, maybe you guys have heard about Phineas Gage, who had the railroad spike come through his head. He had damage to his prefrontal cortex, right? And then he had trouble making decisions and planning for the long term. Okay, so the prefrontal cortex poses a particularly difficult problem, but it's unfortunately also really important. So maybe if the prefrontal cortex never really came up in disease, then you could be like, all right, maybe it's just unique to humans, or it's special, or it's different, and we don't really care. We'll just kind of ignore it. But the prefrontal cortex seems to be at the center of most psychiatric disorders. There is not a single psychiatric disorder out there that does not involve some abnormality of the prefrontal cortex. There are other areas that get involved too, but the prefrontal cortex is always there. All right, so the prefrontal cortex has a whole bunch of different components. And right now I'm going to focus on two of them. One is that anterior cingulate area. That is an area kind of in the middle part of the prefrontal cortex. And the orbital frontal cortex. The orbital frontal cortex is the part of the prefrontal cortex that you would see if you could look straight up from your eyeballs. So it's this part that's like right here. And so I'm just going to focus on those two areas for now. All right, so to give you a sense about just sort of the range of opinions, I want to give you some sense that when it comes to the similarities between orbital frontal cortex and anterior cingulate cortex in rodents versus primates, that this is not a solved problem, and it is something that people argue about a lot. So some people think that the entirety of the primate orbital frontal cortex is somehow represented just in the lateral, meaning just the stuff off to the side of the orbital frontal cortex of the rat. Some people think that only the back parts of the primate orbital frontal cortex are present in the rodent, which would mean that all of the front part, middle parts, you couldn't model those in a rodent. You just have to throw up your hands, right, and give up. As far as the anterior cingulate, some people think that the rodent anterior cingulate is most similar to the lateral prefrontal cortex in primates, which would be a pretty major shift, actually. And others think that it's similar to this, to similarly placed topographically regions in the monkey and the human. All right, so you could imagine, okay. So the prefrontal cortex is at the front of the brain. The rodents have a front of the brain. There's stuff at the front there. You could say, okay, there's stuff at the front. Let's try to carve it up and figure out which parts are most similar to which other parts of the human brain. And you could imagine a number of ways, a number of criteria that you would use to do that. And other people have used those, like how the different layers of cells arrange themselves. You could try to use function, which is kind of what we're all interested in anyway, but then differences in behavior across species cause problems trying to figure that out. So, and I could list more. You could try to figure it out using development. You could try to figure it out using sort of neurotransmitter profiles and so on. But the approach that we took was to try to use anatomical connectivity. So remember back, I started out by saying anatomical connectivity is the wiring diagram of the brain. So we wanted to, because anatomical connectivity is something that I can study in rodents and something that I can study in non-human primates, and using the same methods, and I don't have to try to match their behavior or anything. And then going from the non-human primate to the human is actually not a much less tricky problem, and other people have sort of worked a lot of that out. And so once we've made the leap to the non-human primate, we can sort of make inferences about the human. In this case, rats are a relatively easy species to do track tracing in, but we can also use what they tell us to learn about mice. And so, okay, so how would we think about anatomical connectivity for figuring out similarities and dissimilarities across species? Well, we want to look at the pattern of projections to a conserved region, meaning a region that we're sure is pretty much the same across the rats and non-human primates. And the region that we chose for that was the striatum. All right, what is the striatum? You guys have probably learned some basic basal ganglia circuitry. And so the striatum is the gateway to the basal ganglia. So here's the striatum right here. Recall that the cerebral cortex, the entire cerebral cortex, with one exception, except for V1, projects with the excitatory transmitter glutamate to the striatum. And then the striatum projects via the indirect pathway to the globus pallidus external segment, via the direct pathway to the globus pallidus internal segment, and then to the thalamus, and then back to the cerebral cortex. And this circuitry, the basal ganglia circuitry, is what goes wrong in Parkinson's disease, and is what goes wrong in Huntington's disease. But it's also what goes wrong in a lot of these projections from the cerebral cortex to the striatum are abnormal in psychiatric disorders like obsessive compulsive disorder, post-traumatic stress disorder, bipolar disorder, depression. All of these show abnormalities in the projection from the prefrontal cortex to the striatum. Okay. But the important thing about the striatum is that unlike the prefrontal cortex, the striatum, even though it's shaped differently in different species, it has all the same components. It's kind of an older part of the brain. It's subcortical, and it has this kind of organizational structure in both rats and non-human primates. And so we're like on much more solid ground with the striatum when we're looking across species. So we're going to try to leverage the fact that we know so much about the striatum to learn more about the prefrontal cortex. All right. So what we did was to do those injection sites. Remember, I showed you we put dye all over the brain, injected dye in different parts of the brain. So we injected that in the rat and in the monkey. And what I'm showing you here are 3D renderings of the injection sites, which are shown in black, in different areas of the rat and monkey, anterior cingulate cortex and orbital frontal cortex. Now, there are very few people that look extensively at both rat and monkey brains. And so I'll tell you, depending on which audience of neuroscientists I'm talking to, they have no idea what this is, or they have no idea what that is. Full professors, they have no idea half the slide. Nobody ever looks at both brains with very often. So I'm going to walk you through this slide, and the same way that I walk them through that slide. This is the rat anterior cingulate cortex, and it has sort of three major components. CG, which is up here, it just stands for cingulate, but it's very confusing because the whole thing is technically cingulate, because people are bad at naming things, and I'm sorry, and it's not my fault. And the PL, which is the prelimbic cortex here, and IL, which is the infralimbic cortex here. Oh, I should have said, this is a sagittal section, so it's as if you sliced your brain right in half between the two hemispheres. And this white stuff is the corpus callosum. So this is like a classic sagittal middle section. And so we placed injections all over the CG, the PL, and the IL. Then this is an orbital view. This is the front of the brain. This is toward the back of the brain. This is the middle, and this is the outside edge. This is lateral. And so really the feature of the orbital frontal cortex that we're interested in is the medial part versus the lateral part. These are the same views in the monkey. So this is a sagittal view. This is the corpus callosum here. And the monkey has three different components: area 24, which is that dorsal anterior cingulate cortex that I mentioned with post-traumatic stress disorder; area 32, which sort of curves around here; and area 25, which is down here. And similarly, on the orbital frontal cortex, we have medial versus lateral. All right, so I said the striatum is relatively conserved, but I also said that it's shaped differently. So how are we going to get started here doing these analyses? So within the striatum, there are some extremely conserved features and some features that are really easy to identify across species. One of them is called the shell of the nucleus accumbens. So the most ventral part, meaning the part that's closest to the bottom of the brain of the striatum, is called the shell of the nucleus accumbens. And you can just see it by staining it. You don't need to do anything special. So I can see it in a human brain. I can just do a stain. It shows up very, very easily. And so we'll start there. So what I did here was to create, was to color each injection site according to the proportion of its projection to the striatum that falls inside the shell of the nucleus accumbens. And what you can see is that in both species, there's sort of a hot spot that stands out. It's the infralimbic cortex here in rat, and the area 25 here in monkey. And those are really the parts of the brain in both species that have the strongest overlap with the shell of the nucleus accumbens. So we did sort of a number of analyses of that type. And what we created were these connectivity profiles. So now I've showed you the prefrontal cortex. I'm actually showing you what the striatum looks like in both species. And I warned you that it has different shapes. So over here, these are drawings of the rat striatum, and these are drawings of the monkey striatum. And we will actually get to why they have such a different shape, but for our purposes right now, it doesn't really matter. So in each case here, the colored region is the projection from different parts of the anterior cingulate cortex and the orbital frontal cortex. So we made these connectivity profiles, and we looked at their overlap with the shell of the nucleus accumbens and other conserved features of the striatum. And so first I want to focus on the orbital frontal cortex. So what we found is that in both the monkey and the rat, the orbital frontal cortex has a specific projection zone when it's coming from the medial orbital frontal cortex versus the lateral orbital frontal cortex. Meaning medial versus lateral orbital frontal cortex are different from each other, but that difference is maintained across rats and monkeys. And then we wanted to look at the anterior cingulate cortex, and that's where things got trickier. So I already said that the infralimbic cortex is very similar to area 25, which in monkeys, but then things got a lot harder when we considered the prelimbic cortex and CG versus area 32 and area 24. These weren't perfect matches. And so especially when we thought about area 24 here, it just seemed like it occupied a much bigger part of the striatum, including getting into the nucleus accumbens, into that shell, into that very ventral zone, than CG allowed us to, and more dorsal and lateral than PL allowed us to. So it didn't seem to have a perfect match. And so we looked at different subcomponents inside of area 24 in the monkey, and what we found was that the more rostral parts of the area 24 indeed didn't seem to have a close match to the rat, but the more caudal parts sort of did. So the more caudal parts are more similar to CG, and it's the rostral parts that look quite dissimilar across species. So it's possible that the rostral part of area 24 is just poorly matched in the rodent, and that's just okay. That's just, we just have to assume that maybe there was some change over evolution. And so the whole goal of this experiment was to create a sort of guide like this, a map like this, so that if somebody comes to me and says, all right, there's hyperactivation in area 25 in humans for depression. There is, that's a true fact. If I wanted to model that condition in rats and then try to treat it somehow, where should I go? And the answer is to infralimbic cortex. Let's say that you had a finding from rats, let's do the reverse order, that projections from the medial orbital frontal cortex in rats were important for establishing goal-directed decision-making, whatever, any number of things. Where should you go in the monkey or the human? You should go to the medial orbital frontal cortex. And I know that sounds like it should be obvious, but it wasn't obvious. Remember back, I said there were some people that thought that the entire OFC was encapsulated in the lateral part of the rat. But then, of course, things are a lot harder when you're talking about these other anterior cingulate areas, such that the rostral area of 24 doesn't seem to perfectly match one or the other of prelimbic cortex or CG. And so what we hope is that this approach, which we're bringing to other parts of the prefrontal cortex and other parts of the cerebral cortex more broadly, will allow us to translate results from rodents to non-human primates to humans and then back again. And I also think that there's an interesting argument here, and I want you to stay with me because it's a little bit tricky. So you could interpret this two ways. You could say that striatal connectivity is indicative of broader similarity, and that what I've showed is that, say, infralimbic cortex is similar to area 25, period, dead stop. Alternatively, instead of thinking about regions of the brain, we could think about pathways of the brain. We could think about connections of the brain. So that instead of talking about the infralimbic cortex as a region, instead maybe I should say infralimbic projections to the striatum, because there are some neurons in the infralimbic cortex that project to the striatum and many, many others that don't. There are some that project to the thalamus, there are some that project to other areas of cerebral cortex, and maybe each of those is unique. And so maybe if the fundamental way that we divide up the brain, which has mostly so far been about regions, ought to be replaced by thinking about circuits or particular connections. And that might sound a little bit wild, but because of the advancements in optogenetics and chemogenetics and all of these fancy tools, viral tools in rodents, I would say at least 50% of sort of of neuroscientists that work in the rodent world are kind of already thinking that way, and strongly distinguish between cells in a given region that project to region X versus region Y. Increasingly that's sort of known as circuit neuroscience or circuit breaking. And I would say that's a major feature of the field that's evolved over the last 15 years as technologies have evolved. And so that leads us to, what about humans? monkeys, sometimes you can sort of do optogenetics in them. What about, but really we're interested in solving problems for humans. Is human neuroscience just left behind? Because all of those tools that I talked about that can manipulate specific connections and interrogate their functions, those are just not available in humans. They're too invasive. It's just not possible right now. So does that mean that we're just out of luck? And so some of you might have seen pictures like this. This is an image from a diffusion MRI scan, so diffusion magnetic resonance imaging scan. And it's a type of non-invasive MRI that was developed in order to, among other things, try to advance the study of sort of human circuit neuroscience. So I'll tell you what it looks like. This is an MRI machine. Many of you will have had or attended clinical MRIs. A typical clinical MRI, you measure how strong an MRI machine is by its field strength, by its number of Teslas. And so a typical clinical is 1.5 Teslas. Or if you're doing something really fancy where they're trying to maybe look at a location, a particular location in your brain, they might put you at 3 Teslas. This is a 10.5 Tesla MRI. It's the strongest large bore, meaning a human can fit inside it, strongest large bore MRI in the whole world. It's the only one in the whole world, and it's here at the University of Minnesota. And in fact, there were major ways that MRI is used that were invented here at the University of Minnesota because we have such a strong MRI team. And so that's sort of a cool thing about Minnesota to know, is that the history of biomedical research in the state is strongly linked to these amazing discoveries that are really important for psychiatry and neuroscience now. All right, so somebody goes inside the scanner and you run a series of sequences. You can do a number of things in MRI. You can try to look at activity, something that's correlated with activity. You can try to, you can just look at the structure of the brain. But there's a sequence that allows you to ask what the diffusion of water molecules is doing in your brain. All right, so why would you be interested in the diffusion of water molecules in your brain? Stay with me. Remember that axons are encased in myelin sheaths, and axons that are traveling from one region to another sort of all travel together. And so when water molecules are diffusing around those myelin sheaths, they're sort of stuck. They kind of just, bounce between those myelin sheaths and travel along the parallel with the myelin sheaths. Whereas if you can picture the arrangement of cells in gray matter, just like without much myelin around it, the water molecules will just kind of bounce all around randomly. So in other words, the diffusion MRI lets you distinguish between water molecules that are lined up along myelin sheaths, sort of pointing in a particular direction, versus bouncing around. And so it points where the axons are pointing. And that's really, really cool, because the way that neurons communicate information from A to B is by sending axons from A to B. So maybe if we could march along the water molecules, march along where they're pointing, we could know what the connection is from A to B. And so remember, at the beginning, I said this is important. We don't know the wiring diagram of the human brain. That's still true even with this method, even with diffusion MRI. But we can try to get there. The major problem is that an axon, a typical axon, is very, very small. It's under 1 micron, just under 1 micron. The typical resolution for an MRI machine, meaning what it can look at, is about a cubic millimeter of space. So that's just a complete, this is orders of magnitude apart. So this isn't going to let you look at 1 axon. It's not going to let you look at 2 axons. It's not even going to let you look at hundreds of axons. It's looking at thousands to millions of axons per voxel. And so that's sort of an issue, and that means that it makes mistakes. And those mistakes mean that it doesn't perfectly capture the anatomical connectivity of the brains where we've looked, within species to try to figure out how things work. And so to try to figure out connectivity. And so what this means is that as we've tried to use diffusion MRI and tried to advance diffusion MRI to understand human brain connectivity, the stuff that we usually cared about, so this is gray matter, right? We usually think about the gray matter of the brain, and we're interested in what the cell bodies there are doing, has been replaced. So we don't usually care about the white matter of the brain where all those axons are, but suddenly we really, really need to. Oops. All right, so I kind of alluded to this, but I just want to hammer it home. So in order to estimate connectivity from diffusion MRI, you perform what's called diffusion tractography. And the tractography asks, so let's say you want to know connectivity between the connectivity of this voxel, so this particular tiny part of the brain. Well, you're asking which way are the axons pointing in this voxel? Okay, the axons are pointing along this axis. See, the arrow is pointing this way. And so the algorithm marches from this to from this voxel to whichever voxel the axons are pointing to based on the diffusion of water molecules. So it's pointing here. Okay, so now which way is this voxel pointing? It's pointing, it's pointing in this direction. All right, that means we'll go down here. All right, what about this one? Oh, this is pointing this direction. Now we'll go down here, and so on and so forth. So that eventually you come to the conclusion that this voxel connects with this voxel, but not with this voxel. Okay, that's kind of how diffusion tractography works. And all of this is taking place inside this stuff that we didn't used to care about, this white matter. All right, so that's diffusion tractography. That's how it works. And just to say, because of that major disconnect between 1 millimeter cubic voxels and axons that are less than a micron, when you look within species and look at connectivity as measured through the injection of dye, like I talked about, versus using diffusion MRI, you get a positive correlation between those two, but it's not strong. And so what that means is that diffusion tractography is making some errors. And what we'd like to do is try to correct those errors and eventually know that we've corrected enough errors to get a much more reasonable estimate of connectivity of the human brain. All right. So I want to talk to you a little bit about one particular feature of the white matter, one particular bundle in the white matter, and it's called the anterior limb of the internal capsule. And so it's this stuff right here that's actually, this is the striatum. So you've already seen it in the last section, but the anterior limb of the internal capsule has very little to do with the striatum. It just happens to bisect it. So it happens to run through it, kind of like a highway that runs through town but doesn't stop. And so the anterior limb of the internal capsule brings fibers from the prefrontal cortex to deeper structures like the thalamus and the brainstem and the spinal cord. Okay. All right, so the pipeline that we've developed to try to get all the way to diffusion weighted MRI in humans, all the way to connectivity in humans, is to take the track tracing in monkeys, that injection of dye with the method that I've already been talking about, and add in another step, which is diffusion weighted MRI in monkeys. The idea here is, for a long time, people were just doing track tracing in monkeys and then diffusion weighted MRI in humans. And then when they didn't match, they were kind of stuck. They said, well, human brains aren't the same as monkey brains. That's true. What do we do? How can we tell whether this is a species difference or a problem with diffusion weighted MRI, diffusion MRI? And they were just sort of like, we don't know. What should we do next? And so what we've done is to add in diffusion MRI in monkeys. And so what we can compare that to the gold standard of track tracing in monkeys and know, is this difference because of a problem with diffusion MRI, or is it a possible species difference? And spoiler alert, most things that we find where there's a difference between track tracing in monkeys and diffusion MRI in humans, we find that the diffusion MRI in monkeys matches the diffusion MRI in humans, meaning it's a problem with the method, not a species difference. And so what this looks like is we put injection sites in different parts of the prefrontal cortex here. So all that's important here is that green is above yellow is above red, and that topography of their fibers, of their axons, is maintained in the anterior limb of the internal capsule, where green is above yellow is above red. That's true in diffusion weighted MRI monkeys. Green is above yellow is above red. And same thing in the anterior limb of the internal capsule. And then the same thing in the humans. Green is above yellow is above red. All right, nice. This is an area where, this is an example of where diffusion weighted MRI worked very nicely. And we're pretty confident that these connections in humans are real. But that's not always the case. Sometimes there's a mismatch. So this blue area is pointing to nothing. That's because there's nothing there. There's not supposed to be anything there. But when you do diffusion weighted MRI in monkeys, you get something there that you shouldn't. And this leads to bad connections. And then you get the same thing when you get to do diffusion weighted MRI in humans. And so we know that's just a bad connection. It happens. I could go into the details of, we're pretty sure we know what the problems are there that are causing that, but we can just tell the algorithm, don't do that. That's bad. Like, don't do it anymore. And by the end, once we've built all of that up very carefully, we can create a map of the different regions that are the different connections that are passing through the anterior limb of the internal capsule. So this is a map. The different colors are different prefrontal cortex brain regions that are sending axons through the anterior limb of the internal capsule. And the cool thing is that if you ask, for example, are there abnormalities in any part of the anterior limb of the internal capsule, and what might that mean? Well, we already knew before doing this work that there was an abnormality in a particular chunk of the anterior limb of the internal capsule. In this case, in bipolar disorder patients, it was right there. And now that we have made this map, we know that those are projections from the ventrolateral prefrontal cortex to the thalamus, and that those are the connections that are problematic in bipolar disorder. What about the rat? There's no way that I could go through this without at least mentioning the anterior limb of the internal capsule in the rat. Well, I showed you this bundle in the human. Actually it was in the monkey, but it's the same in the human, is like this nice, well encapsulated, well-defined bundle that goes through the striatum. And remember back when we were talking about the rodent monkey project, I said they have different shapes. Well, that's because in a rodent, there's no such thing as like a well encapsulated anterior limb of the internal capsule. Instead, it has all these tiny little spots, tiny little spots that are called fascicles. And the fibers that are traveling between the prefrontal cortex of the rodent and the thalamus and brainstem, so the similar connections run through those fascicles. But are they organized in any way? And we were able to show that indeed they are organized, just on sort of a tilted axis from what we learned from the monkey and the human. And so now we sort of have a map of the anterior limb of the internal capsule in the rodent, in the non-human primate, and in the human. And that's kind of cool because not only because of what I showed you for bipolar disorder, but the anterior limb of the internal capsule is a target for neuromodulatory treatment for obsessive compulsive disorder and depression. And that means that people do stimulation of those regions in very severe forms of those psychiatric disorders, which I'm happy to talk more about because it's a very interesting topic later. And that's sort of an area where we need to have the model species in order to advance that treatment. Here's another quick example of where white matter bundles are interesting and how we can divide them up. This is a white matter bundle called the cingulum bundle. So I'm showing you here again, a sagittal slice is if you cut the brain like this, and you're just looking at the middle of the brain. And this dark piece is a 3D rendering of the cingulum bundle. And it connects the frontal and parietal and temporal lobes of the brain, all the middle parts. It's just a great big white matter bundle. What we did was to figure out which parts of the brain it was connecting. And then remember, I told you that the area 25 in humans was hyperactive in depression. Well, people at Emory developed a method where they do what's called deep brain stimulation. They stick an electrode in that area in people with very severe depression, turn it on, and then they seem to get better, which is sort of amazing. And for a while, they thought that they were targeting the sort of gray matter of the cells right there. But eventually they learned that they needed to target specific sets of fibers that were in the general area of the electrode. And one of those was the fibers of the cingulum bundle. And so knowing which fibers were present near there and how to make those and where to put the electrode to target them became a very important problem. And so these works sort of advanced together. I'm going to skip over this. And so I hope I've convinced you that when thinking about connectivity and investigating connectivity can help us provide a bridge across different ways of looking at the brain using, say, non-invasive neuroimaging like diffusion MRI, and using a more circuit-based invasive methods in non-human animals, providing that bridge across species and providing a bridge, a sort of translational bridge to disease from basic neuroscience. And with that, I'll thank my lab. I have a big lab with a number of students whose names you can see here, and collaborators at the University of Minnesota. A bunch of this work was done when I was a postdoc at the University of Rochester with Suzanne Haber. And you can find out more on my lab webpage right there. Thank you.
Thank you very much. Okay, that was wonderful. And I know that hold on, I'm trying to navigate how to deal with Zoom here. Just give me a moment.
Do you want me to unshare or so that we can talk, or do you think people will want to see slides again?
Why don't you, how about, can you unshare, and then if we need to pop up slides, you can just reshare. It takes a little more effort on your part. Sorry about that, but
it's nice to see people's faces sometimes.
I know. I was just going to also we have this fancy hybrid set up in our classroom, and so I'll kind of turn it so you can see. I didn't fill you in, but we have a system where half of our students are in the classroom at any given time and half are on Zoom, and so we kind of make this work. So thank you. That was a really, really nice presentation, and I know for at least the neurobiology students that ties in really well with a lot of things that we've talked about in the past 3 weeks. So we're on a block system. It's been like a very intensive kind of neurobiology situation here. Okay. So at this point, let's open it up to questions from anyone in the audience. I think first, it makes sense to talk through specific questions about the work that Dr. Heilbronner was presenting in her talk. And then, kind of as we talk through a lot of those questions, I know there are other questions that the group has for you, kind of about science more broadly and grad school and all that kind of stuff. Does that work? Okay. So I will open it up. What questions do you have for Dr.
Heilbronner? I can start. So I just have a quick question. So you discussed how with this study you created a roadmap for non-human primates and then rodents. Mm-hmm. Has there been any attempt to create that between other species that can be used in research?
That's a really interesting question. So when we do other species can offer certain advantages, like birds, for example. Birds have an amazing capability to learn bird song, right? And much of that learning process seems to mirror our language learning process. And I think the main species used there is zebra finches, although I might be wrong. Flies, of course, have their own unique capabilities, things like that. When we go, we need a lot of information, not just this, but a lot of information about the organization of the brain in order to use a species as a model species. And so neuroscientists kind of like to lock in to one species or the other. And so that's part of why mice are used so much, and then rats, and then sort of macaques, marmosets sometimes, like I said, flies, and zebrafinches and zebrafishes are also used because of their translucent properties. But even when it might be better to use another species, it is often going to be very challenging because you will know very little about their brain. And so there have been approaches, so nobody's really taken a deep dive into the prefrontal cortex of a drastically different species. There are other monkey species that people have fussed around with about the prefrontal cortex, and of course mice. So this study was in rats, but people have done things with mice. You know, we know something about the organization of the prefrontal cortex in cats, for example, in pigs, in sheep. We don't know as much. I mean, I was really trying to subdivide the prefrontal cortex, so we might not know as much about those. So the answer is sort of yes and no. And so there's definitely a large contingent of neuroscientists that would like us to expand the number of species and use the best species for the question. But unfortunately, sometimes there hasn't been the background, the groundwork. Like not every species has a brain atlas. So how would you, like, where would you would have to make that from scratch, you know, if you wanted to start with a novel species. So, you know, I can see it from both sides. Yeah. So if you're interested, the people that use, that really take advantage of species differences to ask questions about the brain are called neuroethologists. So like ETH, neuroethologists. And that's a fascinating area of research. Yeah.
Dr. Heilbronner, I also have a question related to the species that you use.
So this is, you might want to, can you like raise your hand so she can see? Yeah, there's
Yeah,
Dan right there.
Yeah, in the middle of the class.
No, maybe I wonder if I double click, can I get you? No, I can't. I thought I was going to be able to get you large on the screen, but no.
Yeah.
But I guess, yeah, so my question related to the species that you used. Oh, there I am. I guess. So we know, you mentioned why you used the two species that you used, and we also know, as you said, rats and mice are used very often, especially in neuroscience research. When it comes to using the non-human primates though, what challenges exist in acquiring them and working with them? I feel like they would be much more challenging. So
yeah, they are. So we don't use them at the same neuroscientists don't use them at the same frequency as we use rodents, and we do it very, very carefully, so that each, you know, with an eye toward animal welfare. And so there's sort of two main non-human primate model species that we have a lot of background information for: the rhesus macaque and the marmoset. And the rhesus macaque has been used for decades, and we know a lot about its brain organization, especially relative to the human. So you get to, when you use them, you get to build on that. And like I said, I was able to just jump sort of, I don't want to say freely, but with a lot of background information between rhesus macaque and humans because of that, because of the work that's been done in the prior decades. They're only present in certain facilities, at certain schools. There are enhanced regulations. All of that is, to ensure that we're not repeating ourselves, we're not doing something for bad reasons, nothing like that. But they remain a really critical model species for translational work. And for reasons like what you just saw, we couldn't have done this work with any other intermediate species there between rodents and humans. And especially as we investigate psychiatric disorders, there are parts of the brain that are present in the rhesus and the human that are just not present in the rodent, and circuits that we need to be able to investigate. And so, you know, it's not something that I take lightly. And it's something that the community is in constant conversation about. And again, it is done sparingly according to where it needs to plug specific translational holes.
Thank you.
Good questions. What other questions do you all have?
I have a question.
Mhm.
Sorry, I'm a little sick, so my voice sounds weird. When you were comparing the ACC and OFC connections to the striatum, why did you use the anterograde tracer from specifically the ACC and the OFC instead of a retrograde tracer from the like emotional processing network? So the shell and then the hippocampus projection zone and
Oh, I see. I see. Yes, we could have. And okay, we thought about it. That's a really good question. So it sounds like you know this, but just for background, the striatum, the cerebral cortex only projects to the striatum. It doesn't receive projections from the striatum. But what she's asking is more complex. Why not put a retrograde tracer in the striatum and look at the pattern in the cerebral cortex? Yeah, we tried. The striatum, the cortical striatal projection pattern has a lot of overlap in it. And so anytime we were placing injections in the striatum, you wind up getting, and people have known this forever, you wind up getting broad swaths of the cerebral cortex. And it can be difficult to know in advance that, oh, I should target this exact part of the striatum to see really the difference, to carve apart the prefrontal cortex at its seams. Whereas I can really target one part of the prefrontal cortex with an anterograde injection and not have it bleed into the others. I can tell you all of it is, I had a lot of retrograde injections in the striatum in the lab. All of it matched. It's just that it was harder to carve it at its joints going the reverse way because of the overlapping projection, the integrative nature of the cortical striatal projection. It's a really good question. Yeah.
I have a question kind of going off of that. So you focus more on the cortical striatal pathway and like the prefrontal cortex. Are there any other regions in the brain that you think would be important to research for psychiatric diseases?
Yeah, definitely. So just staying with the prefrontal cortex on one end, obviously it doesn't only project to the striatum, it projects to lots of other things. And some of those things are also conserved and we could do similar analyses. So one that comes up a lot is the thalamus. Unfortunately, the thalamus is not as conserved as we want it to be across species. And so that's sort of a classic, thalamic projections are sort of a classic way to think about similarities and dissimilarities across species, but it didn't work great for this. So we sort of tried it and it was too hard to figure out the thalamic organization once you got down into the nitty gritty the way we did in the striatum. As far as other cortical regions, all right, so if you go back from the prefrontal cortex to this part of the brain, to the sort of the parietal cortex, the parietal cortex has its the sort of caudal equivalent of the anterior cingulate called the posterior cingulate. And the posterior cingulate cortex is a region that I've been obsessed with since I was a graduate student, because it's part of a group of regions in the human brain that turn off when you do a task. So if you stick somebody in an fMRI scanner and you look at the activity of their brain and you ask them to do a crossword puzzle or, you know, I don't know, do a math problem or do any number of these psychology tasks that people have human subjects do in the scanner. In general, the posterior cingulate will turn off, which is like fascinating and amazing. And if you ask people to just go into the scanner and just do nothing, it turns on and goes crazy. And so this is like an area of the brain that also is extremely involved in psychiatric disorders. But it presents, just like the prefrontal cortex, this huge challenge across species. In the case of the posterior cingulate, it's more that people haven't paid the cross species problem enough attention. And so that's like a next big target for my lab, is to try to do a very similar study, maybe a little bit broader ranging, not just looking at the striatum, but looking at other conserved areas of the posterior cingulate across rodents, non-human primates, and humans.
Other questions?
I kind of have a methods question for you. So when you were looking at like overlapping projections, like in figure 9 of the paper,
Mm-hmm.
how exactly did you like quantify the percentage of overlap? Like obviously you can see it, but how did you quantify it?
Yeah, there are a number of programs you could use to do it. It's just like the actual area of overlap, right? And so I think that I used some code that no longer exists in like the graphics program I was using. And then my student, now that we're working now that on these next set of studies, has MATLAB code to do it. Like it's just literally like a you know, if you have two circles, what is the intersection of them like? It's not very tricky. Yeah.
I have a question for you. So you talked a lot about the different like technologies and techniques used to like look at these different connections. Would you say that technology is the biggest barrier between further understanding, or what would you say the biggest barrier would be in finding this connection?
The federal government definitely thinks it's technology. So I'll tell you a story, which is that we're in phase 2 of what's called the Brain Initiative, which provides just enormous quantities of money to people doing technology development in neuroscience. I mean, a group of very senior, very, very wise neuroscientists got together and said they thought that was the major barrier, which was that we couldn't record from enough neurons at the same time. We couldn't look at connectivity in humans. We couldn't, any number of things. There's just a lot of things like that. But each technology comes with its own problems. I don't know. A lot of the technologies that to you all seem like they've been around a while, like optogenetics, have only came into fruition when I was a grad student. And so we're just learning, I think that's just not, it's not that long. We're just learning what a major technological advance like that means for our understanding of the brain. In addition to technology, there are conceptual shifts that there are big conceptual shifts happening right now, especially on the human side of things, that I hope will help. But it's also possible they could hurt, right? It's just we needed a change. So I'm not a psychiatrist, but let me, tell you what the sort of impression is and the priorities are from the National Institute of Mental Health. They felt at some point in the last decade like our categories for different mental health disorders might be completely made up and we're not great. And so and psychiatrists know this. They say, okay, somebody with figuring out whether somebody has bipolar disorder or schizophrenia can be very, very tricky, especially across, there's major individual differences, there's differences across populations and so on. Are these biologically sound categories and should they relate to treatment or not? So somebody presents to a psychiatrist, what label do you give them and what treatment path do you set them on? And the sense was that we were not doing a great job with that as a field, and that treatments were being held back in part because of that. And so there's been a shift to try to look at like specific deficits and symptoms and how they cluster, rather than trying to like treat a whole disorder or something like that, to figure out what are biologically meaningful classifications and can we use those to advance treatment. Hand in hand with that is this sense that there haven't been enough major advances in psychiatric treatment. You know, the main pharmaceutical treatments for psychiatric disorders were developed in the 70s and 80s, right? That's not great. There have been advances in some, like especially side effect profiles and things like that, but it just seems like there should be more. And so those two things are moving. Those are conceptual shifts that are aligned with our current funding environment that may also have a big impact. So I would say technologies are really important, but not correct, because we don't know what's correct, right? But being willing to be dynamic in our concepts and in our categories and in our thinking is also really important.
So like in kind of keeping with that like same theme about like treatments and like applications to human disease, like so like how has like your work been like used in connecting like mouse models to primates to humans? How has like that been used thus far to like further our like understanding of like human illness or like maybe even possible treatments
Yeah.
specifically?
Things move a little bit slowly. I wish I could say that a paper published in 2016 would like change treatment by now, but it hasn't. So instead, I'm going to tell you what I think that when we talk about circuit neuroscience, what that looks like as far as treatment for humans. So most things that we, most treatments that we have for mental illness are pharmaceutical or therapy treatments that, you know, you could imagine pharmaceuticals don't target a particular cortical brain region. They target usually in a broad way a lot of neurotransmitter systems, neuromodulator systems. And I think that they have a lot of efficacy for a lot of people, but there is clearly, there would be a lot of value, enormous value, to being able to target a region or a connection. Like I showed you in PTSD, hypoactivation in the dorsal anterior cingulate cortex. Right now, we don't have a non-invasive way to just enhance the activity in the dorsal anterior cingulate cortex in humans, but everyone's working on it. So I think this sense of like being able to target specific regions and specific connections non-invasively in the human brain is a huge area of research. And then all of knowing the connections becomes sort of incredibly important. So right now, what we can do to target regions and connections instead of whole, you know, pharmacological systems is there's a few things. One is very invasive, which is deep brain stimulation, which is what I was talking about earlier. Deep brain stimulation was developed to treat movement disorders like Parkinson's disease. And so it's an invasive procedure where you put electrodes in the human brain, turn them on, and then in the case of Parkinson's disease, the patient often dramatically improves. And so this obviously, you know, happens under the care of both a neurosurgeon and a neurologist, and usually many of them. But deep brain stimulation for Parkinson's disease and other movement disorders, that's in pretty widespread use. And it works. I mean, not for everyone, but it works. Deep brain stimulation for mental illness is obviously more challenging in a number of ways. You could imagine like getting patients, you know, being sure that invasive brain surgery is worth it for mental illness, and knowing that this is something we, even if it's a very low risk procedure, it's still brain surgery. That the ethical challenges there are substantial, but we do it as a field sometimes, and so there are targets for deep brain stimulation, many of which I went over either explicitly or implicitly. So the anterior limb of the internal capsule, which I went over in the second study, is a target for OCD and depression. The cingulum bundle is a target for depression in one spot, and a target for depression and chronic pain in another spot. Those are sort of major areas of research, both because we want to improve the deep brain stimulation and because we want to try to make non-invasive measures that match the changes that happen there. So the non-invasive side of things, you can do stimulation to the outside of the head using transcranial magnetic stimulation or transcranial direct current or alternating current stimulation. And so that involves like putting a paddle on the head and turning it on. And the advantage of that is it's not brain surgery. And it does seem to work. For example, the dorsolateral prefrontal cortex is a good target for depression. This part of the brain, the orbital frontal cortex, like I was talking about, is a target for addiction. And right, so it's not nearly as invasive. The problem is it is also not nearly as targeted. So it winds up targeting a huge swath of the brain. And we're trying to make those smaller and smaller and also go deeper and deeper. And so as those technologies advance, we will hopefully be able to shift psychiatric treatments from being, well, maybe this is a little bit pie in the sky. I want to be clear about where I'm talking about hopes and dreams versus happening tomorrow. I hope that we will get to the point where we can flexibly, non-invasively target brain regions and connections rather than the entire serotonin system of the brain or something. That was a long answer, but you got me on a rant.
That was very cool. Thank you.
I have a question. Sorry, I'm way in the corner.
I'm gonna do my best to
Oops, wrong way. Other way.
Sorry,
Okay,
way in
yes.
the corner over there.
Oh, all the way over here. So, okay, this is kind of more of a broad question, but this article was published that we've been reading about 5 years ago, and I was just kind of curious, since then and now, where has your research kind of taken you? What insights has this given you?
Yeah.
What's happening now?
Yeah. So like I said, I'm trying to apply a similar method to sort of the back part of the brain. In addition, the lab that I was working in, and I was not involved in this study, but it's really cool, took that. So the part of the brain that I said in that study that was most dissimilar across species, the rostral part of area 24, later discovered that part of the brain in monkeys and in humans is, let's call it a hub for connectivity, meaning it gets inputs from more areas than other parts of the cingulate do. And so they've sort of they're sort of chasing that functionally to try to figure out what the hub does. I would like to expand and make sure that it's not in the rodent anywhere. And then one thing that was not in that study that in my opinion is a big glaring hole is the problem of what's called the dorsolateral prefrontal cortex in humans and non-human primates. So this chunk of the brain, which I have pointed to before because I was talking about transcranial magnetic stimulation to that part of the brain. So when you, for depression, this outside part of the brain is not, it may not, we have no idea whether it's present in rodents or not. And it's an area of huge controversy that I just didn't address in that paper. I just didn't answer it. I punted it to a later date. And the reason is because obviously there is an edge of the rodent brain. It's occupied by primary motor cortex and then primary somatosensory cortex. So we know that it's not this stuff, which is in humans and monkeys important for like rule learning and abstract thought and like studying for your test and stuff like that. So there's areas I want to include that and start to figure out whether there's any part of that brain that's of the rodent brain that's there. The other thing that's happened is, I moved to University of Minnesota, I set up my own lab, and the group at the University of Minnesota is very interested in features of psychosis. And that often involves different parts of the prefrontal cortex. And so we have a large interdisciplinary group that spans, so we have people looking at the activity of the anterior cingulate areas in rodents that have different genetic models that may change their synapses in ways that are similar to the changes in humans in psychosis, in disorders of psychosis like schizophrenia and some kinds of bipolar disorder. And then we have people doing psychiatric neuroimaging, doing MRI and diffusion MRI on patients with psychosis. And so it's become urgent to figure out not just whether the projections from the ACC to the striatum are the same, but whether the projections from the ACC to other cortical regions are the same, especially some in the back of the brain, because that's what is different in schizophrenia. And so that's a team that I'm working with to try to actively move back and forth between the two species. There's other things, but that's sort of a sample there. Yeah.
Cool, thank you.
I have a question.
Oh, go ahead.
So your paper mentioned that the boundary of the nucleus accumbens is considered to be kind of like ambiguous and maybe hard to differentiate from the surrounding areas in the brain.
Yeah.
So did you run into any issues using this region because it was kind of ambiguous?
Oh, so sorry, that was unclear. The nucleus accumbens has, all right. The striatum has three parts. There's the dorsal striatum and then the nucleus accumbens. The nucleus accumbens has two parts inside it, so that makes the three parts. The nucleus accumbens, and they're ordered like this. So the nucleus accumbens the dorsal striatum is on the top, and then the core of the nucleus accumbens, and then the shell of the nucleus accumbens. It's really, really easy to ID the shell of the nucleus accumbens versus the core. So the boundary between those two is extremely clear. It's the boundary between the core and the dorsal striatum that's less clear. And so I use the shell of the nucleus accumbens, but not the core. So if somebody says nucleus accumbens, they mean both the shell and the core, but because I couldn't draw a clear boundary across species where the core ended and the dorsal striatum began, I just used the shell. Does that make sense?
So these have been great questions thus far. I know there were also some questions, we can open it up. I know you all had some questions about kind of the process of science and the process of publishing, or the state of research, the state of neuroscience, graduate school, all that kind of stuff. So if you have questions about the research itself or the paper, go right ahead and ask those, but you can also feel free to kind of broaden your questions as well.
Yeah.
I have a question. So as a professor, is there like a lot of pressure to get papers published? And like, if you did want to be a professor and only wanted to do research, would there be more pressure to get things published?
So my job is to publish things. I mean, I teach one class, one semester per year. So my teaching load is extremely low, and that varies across the profession. So at some places you teach more, at some places you teach less, and you just sort of slot yourself in depending on what you like more, how you like to spend your time. My job is to do research, and the only way that people learn what I've done with my research research is for me to publish. And so publishing is, it's kind of the point. So it's not just like an annoying side thing. It's like, that's like the main output of my lab. The other side, and this is important, if you run a research lab, if your job is mostly research, the publishing is the output, but money is the input. And so I have to spend a lot of time writing grants. And so those go to National Institutes of Health and to private foundations and to internal funding sources. But I need to convince people that have money that I have ideas that are worth funding. Those two pieces, the input of the lab and then the output of the lab and everything that comes in between, that is really, it's actually formalized in my contract, so I don't want to say the wrong number, but it's the bulk of my job is that. But there are jobs that are less that, so you can add it. If I were a psychiatrist, maybe instead I would spend half my time seeing patients or something. I don't do that. If I were, I could teach more. I could do different. Every job is configured a little bit differently, and so I think it's actually a little bit hard when you say that you're a professor. That can mean drastically different things across different institutions and then different departments within the institution. Yeah.
Can I expand on that question? So I happen to know that Ellen is going to grad school next year. She wants to do research. Can you talk a little bit about how in academia, research scientists or research associates who are not faculty, who have purely research positions, can you talk a little bit about how they are placed and kind of the expectations upon them and how they're funded and all that stuff?
That's gonna be really subfield and maybe even institution specific. So I'm only gonna be able to talk about what I've seen at Duke, at Rochester, and at Minnesota in neuroscience, right? But that caveat, those positions are, each one is unique and different. So it's gonna be rare to find postings for PhD level research scientists. Wait, I should back up. If you want to work in a lab as like a technician and just sort of run someone else's ideas, run someone else's studies for your whole life, you can do that. And that can be a very stable and interesting job. And that kind of research technician position is, those people are essential. I don't know what I would do if I lost my lab manager or histologist extraordinaire. They are essential to the practice of doing science, but they generally don't design studies. They might have input, but they don't generally design a research program. A research scientist that maybe has a PhD usually won't run their own lab. So labs are generally run by people, by professors who are willing to get the grant money to fund other people's careers and willing to negotiate papers all the way through the pipeline. And then they're given space at the university. So I have the Heilbronner lab has a physical, space at the university. There are some research scientists in my department. I don't currently have any in my lab that have sort of established permanent positions inside somebody else's lab. So they have PhDs, but they don't want to write grants, which is perfectly reasonable. And they might not want to be in charge of other people's careers, also reasonable. But it's very idiosyncratic. When they're talented, they are in extremely high demand. Because they take a huge weight of running the day-to-day operations of the lab off of the professors sometimes. But that, you'd have to have that relationship, and often it evolves out of doing a postdoc with somebody and then realizing that you just don't want to leave, that it's working out really well. But it's not, unfortunately, in neuroscience, a straightforward career path. It can develop, but it would be hard to plan for. Yeah, I wish I could tell you that that's something that that one can plan on, you know, forever, but it's something that you would sort of have to cultivate again, like personally and individually. At least that's been what I've seen. That's been my experience. I would say only about a third of labs that I work with really have that kind of position, and they usually only have one, and it's somebody, you know, that sticks around.
Other questions?
I have one. How long did it take from like really starting to heavily research this idea to the publication of the paper? And like what steps
Oh
were
no.
involved in that?
Oh, that's so scary. When did Jose visit? Oh gosh. I think it was like 2 years of thinking and collecting the data, and then another year of writing and shepherding it through the publication process. But this one required a lot of thinking, because and we had a lot of fits and starts. Like I said, we tried to do the same analyses with the thalamus, and we were like, whoa. All right, so that was time wasted. Then there's a lot of that. Yes, I don't think that that's terribly unusual. A couple of years to think and collect data. Sometimes data might be sitting around. Yeah, publishing, and it's not the only thing I was doing, right? So there were, I had multiple projects, so there were multiple papers that came out, not just me, but my co-authors. So it's not like I spent 3 straight years on this or anything. It was along with other things. But I think that's a not unreasonable timeline. Or publications. There's a lot of variation. I know somebody who published their last paper from grad school when they started their faculty position 7 years after they finished data collection. And so there had been like 3 years of data collection. So it was like 10 years of project, right? But they had done other stuff in the meantime. Like that happens. Yeah, it can sometimes take time for the ideas to germinate, I guess.
I have a question going off of that. What research had you done prior that kind of led to this or related to this?
Yeah, I did undergraduate work in a behavior lab that looked at decision-making circuits, which are cortical striatal circuits, prefrontal cortical striatal circuits, from an evolutionary perspective. So it was like non-invasive animal behavior research. And that was really fun and a really fun thing to do as an undergrad. But I knew I wanted to get deeper into the mechanism. And so I did my grad work on a very different method, but the same circuits. I did decision making in a single unit electrophysiology, meaning I was recording from single cells as animals were performing decision-making tasks. And I was correlating that activity, those action potentials with the behavior that the complex behavior that I was observing. That was okay. I liked my lab and all of that, and my grad program was good, but electrophysiology was not my game. Like I just, I didn't like, so I'll be honest, I didn't like how uncertain it felt in the analysis domain. Like I felt like I could cut I could do 1,000 different analyses and still not be totally sure what the neurons were doing. And it just drove me crazy, that level of uncertainty. And people that are good at electrophysiology are okay, not only okay with that level of uncertainty, but enjoy the creativity that allows them to work with that kind of extremely rich data set and play with it in different ways. And that just wasn't me. And so I liked the decision-making aspect of it. I really liked the particular circuits that I was interested in. And then, when I considered postdocs, I thought that it would be best to stay in those circuits, but try a change in the method. And when I sat down and looked under the microscope with my advisor, there was a certainty to it. I saw these labeled cells and I said, oh my gosh, they're there, they're real. I know that that is true. And that level of like, of confidence in what I was seeing just was deeply satisfying to me. And so I have this hope that this is out there for everyone, that the particular research agenda and the tools and so on, that there's like a perfect match for everyone. And so I stayed in anatomy in order to sorry, you're on mute. Okay. In order to continue to feel that. I will say, anatomy then comes with its own challenges. Explaining to people why this needs to be done, and because I don't work with behavior directly, can be challenging, and people, it's a smaller subfield, but it's one that I'm committed to and that I like a lot.
Other questions? Megan, who is right here in the corner, so. She's
I was wondering if you could talk more about like what postdoc entails, and then how did you become acquainted with Dr. Haber?
Yeah, so that's actually, I will share with you the honest answer, which is that I met my husband when I was in graduate school. He was also at Duke. He was a postdoc when I was a graduate student, and then he interviewed for faculty positions and got a couple, and then we went and I looked at various postdoc advisors at those institutions. And so we made the decision to go to Rochester together, and there were a number of good neuroscientists there, but Dr. Haber and I, like I said, I sat under the microscope with her at my interview actually, and I just loved it. And so I decided to try to stay in the same city as my husband and try out that postdoc. Now it wound up being very fortuitous because like I said, I loved the field, and she was a really incredible mentor, and we were able to do a lot of really great work together. And then I was able to get my own faculty job independently later. I think you also asked about postdocs in general. So in neuroscience, unfortunately, everyone now does a postdoc, and why that is, I could, opine on for forever, and everybody sort of hates that the field has gone to this, but it just is what it is. And so with very, very limited exceptions, you go from graduate school to postdoc, and it's still considered a training position, but there's no degree associated with it. You do get paid better than a grad student, but still not great. And generally lasts, in the 4 to 6 years range. And so that's kind of a long time to train before people will look at you seriously on the research faculty job market. I couldn't speak to what it would take to get a teaching faculty position. That you'd have to talk to somebody else about, because I am uninformed on that topic. Yeah, I enjoyed my postdoc because I didn't have to take classes. I just did research all the time. And Rochester was an inexpensive city to live in, so it was fine to be there, and on a postdoc salary, plus my husband's. And for me, that was a really good time. But it does present certain challenges, the fact that we've created as a field this need to have this extended training period. So a lot of people switch a little bit their research focus between grad school and postdoc as they learn more about what they like and don't like I did. And then usually their faculty job more closely, the research lab they set up as faculty more closely aligns with what they did as a postdoc.
I have a sort of general question since people are talking about graduate school. What do you think is one of the most important things for undergrads to know going into a graduate program, or like looking at different ones?
Oh,
sort of broad, sorry.
Yeah, I've got to think about that. I think there are kind of two ways that people apply to grad school. And I think it's, important to know yourself to know which category you fall into. I had a particular set of projects that I wanted to do. And I looked for advisors that would do those. Even though, maybe you guys already know this, almost all neuroscience PhD programs now involve rotations, meaning you come in your first year and you're not admitted to a particular lab, you're admitted to the program. And you spend a couple of months in Professor X's lab, and then a couple months in Professor Y's lab, and a couple months in Professor Z's lab. And you do that 3 or 4 times. And then at the end of your first year, you choose a lab in which to do your PhD. So those rotations to me, were interesting, but I knew whose lab I wanted to join, and that didn't change. And then nothing changed there. And so I chose the programs on the basis of the particular lab that I wanted to join at those schools. And that's how I applied to those labs. And that's how I picked the programs. But plenty of people have just more general interests. Like maybe you're just interested in sort of addiction neuroscience. Maybe you're interested in computational neuroscience. Maybe you're interested in, neurodegenerative diseases. And those will be represented in lots and lots of programs. And then what I would say is that so if you're not looking at a particular advisor, it's going to be harder to evaluate because the programs have a lot of similarities, and they're all sort of offering coursework, and they have the rotation system, and maybe there's some degree of cohort bonding and whatever. And so really the best thing to do is to try to figure out, as you're looking through people's, through potential advisors' web pages, are there multiple people there whose research excites you, A, and B, do the graduate students that are currently there seem happy? That can be really important. And as much as you can dig around the weekends, so usually you apply to a program, and if you make the short list, then they will have you out for an interview weekend. And so in COVID times, this has all been over Zoom, and I think that's been really challenging for applicants, and I feel bad for them. But hopefully by next year we'll all be in person again, and you can talk to current students and try to figure out whether they're happy or unhappy. Beyond that, I think that as you're working toward the point of getting to where you want to go to grad school, the important part, you want to be clear, this graduate school is not, so getting a PhD is not taking classes. Like there are classes at the beginning, but really that's not the bulk of your PhD. And if you get A's in your classes, but have no papers at the end of your PhD, you're going to be a much worse off than a student that got B's in their class, B's or C's in the classes, but has papers, right? So it's a really different system than undergraduate. And so you kind of want to be aware of that going in, and not just pursue it because you like taking classes in neuroscience. Ideally, you like the research in some way, and you have some way of knowing that. Long answer.
Good questions. What else would you like to ask?
I have a question.
Oh, hold on. In the classroom first, and then was it Alex? So Alex, you're next. Okay.
I was just wondering, kind of generic question about your research, but have you found anything like really surprising or unexpected in it?
Oh, surprising or unexpected. Let me think about this for a second. A lot of anatomy isn't quite that way. It's more like we build up over time what we have already seen in the literature. But when I was doing electrophysiology in graduate school, remember that part of the brain that I said turned off when you do tasks? Well, I purposefully turned it off during a task using by injecting a drug. And a lot of the theories in the literature said that turning off this part of the brain would make you better at learning things, because you would be less distracted. And instead, the monkey got worse at learning things. And so that was maybe a little bit surprising. But that's maybe one of the things about anatomy. You stare at the tissue for a long time, and then eventually, with insight, the functional story comes to you. So I don't know that it's really a field that that lends itself to surprises, but I don't know. Yeah, it's maybe more like putting together a puzzle.
Alex.
Okay, so have you always known that you wanted to do research, or did you consider other professions, whether that was in neuroscience or not?
Let's see. I did not like science when I was a high school student. I did not, I just didn't care for it. I didn't think that it was interesting. I thought that I would be a lawyer or a politician when I went to college. And then I met a lot of students that were studying to be lawyers and politicians. And then I did not want to do that anymore. No, that's a little bit of a joke, but it's a different personality type, I think, than I had. And I think what happened was I joined a lab my freshman year of college and learned that science research is really fundamentally different from your high school science class, right? And hopefully you guys, I definitely also got a taste of that when I was in, science seminars and so on at the college level, that discovering things is different than trying to make a reaction work with chemistry equipment that's 20 years old or whatever I was doing in high school. And so no, I didn't always know it. And I wasn't like a kid that liked science, but once I started volunteering in a lab, it was just really enjoyable. And I just kept wanting to do it. And I don't think, let's see, I've always thought about backups in case, I didn't get into grad school or didn't get a postdoc or whatever, at each stage, just in case, but I never really seriously went after any other professions.
Thank you.
Mm-hmm.
So I have a question. It kind of seems to me like with each subset of research, there is kind of a development of a internal kind of community of researchers that kind of work together and collaborate together. And so I was wondering, if you had any advice for younger researchers, how to become an active and kind of good member of that community and how to get into that community.
Oh, yeah, those communities exist with multiple rings, right? So I have a lot of internal collaborations at the University of Minnesota. Those are I have people I talk to multiple times a week. I have people that I talk to, a few times a year, all at University of Minnesota. We might share funding, we might share supplies. Those we developed by running into each other at seminars mostly, which is why COVID has so disrupted that kind of work, or that. Then another ring would be the people that investigate similar questions as you, maybe, or the people that use similar methods as you, and then some combination of those. And those people you meet at conferences. And so conferences are a really, and also summer schools as graduate students, there are advanced summer schools, maybe they last a month and you all study one very detailed topic or something. Yeah, we go to conferences. The Society for Neuroscience is a 30,000 person conference that runs every year except for 2020, and alternates among San Diego, Chicago, and Washington, DC. And we go every year, and I send my lab every year, and traveling to that and presenting our work at that to the other 30,000 neuroscientists is a really high priority. We read papers, and when I read papers, I'll sometimes write to the authors or engage with them about their papers at scientific conferences. A more recent thing, and I'm still sort of torn about it, I am on Twitter. Science Twitter is extremely active. it's a sort of weird and slice of science to watch. It's not not entertaining, but it's not 100%. Excuse me. It's not 100% an accurate reflection of the field, but that's another way. Some people are able to really use that kind of networking to their advantage and learn a lot about the field that way and make a lot of connections. I would not say I've done that yet because I just don't know what to tweet, but anyway, yeah, people definitely do it.
I have a question kind of relating to funding. So
Yeah.
Gretchen asked about kind of the limitations of technology and stuff. I was wondering if you ever found it difficult to receive funding or if you've ever had to change your kind of research and methods based on a lack of funding.
I haven't yet. You know, everybody complains about grant applications, but for a lot of us, they're a way to solidify our ideas and get them on paper before we start. So that, you know, when I have had an idea, writing the grant has been more useful than anything else. There are ideas right now that are, I think, good ideas that are as yet unfunded because they, you know, they didn't pan out when they weren't selected. And I haven't done those because it takes real money to do this work. I can't just kind of like do it on the side. But that doesn't mean that I've abandoned them. It means we're constantly discussing them and retooling them and trying again. So, you know, the funding situation is not all or nothing. It's do you have, you know, it's money is fungible. You know, you could have $50, you could have $100, you could have $150, right? Like right now, I've had enough to do the work that I want to do and to have the lab the size that I want. To me, the biggest downside of the way that the whole funding thing is organized is the constant worry that I have that if I don't get enough funding, then I'll have to let people go. And so the way that I try to mitigate that is to be really honest with them about where we are funding wise and how secure their jobs are and for how long, because I can't promise a lifetime of security for my employees. I can promise the cycle of a grant. So when I get a grant, I tell them you're X% on this grant, and that means you're covered for this time. And I think but that's true across all of academic science. And so most people in the system understand that. Yeah, I haven't had any issues yet. But people go through stages of their career where the funding dries up. They have, you know, there's just a lot of randomness in the system. And often universities will step in and help them get through that time as long as they're still trying and so on. Yeah, I'm not going to pretend that it's the easiest system in the world, but it is the system that I was brought up in. So this isn't like a surprise to me. Like all of this was obvious to me during graduate school and as a postdoc, and even as an undergrad, you know, I worked in a lab. And so I could tell that this was like a priority and something that helped, you know, something that would take my time as a professor. So, but like I said, I kind of enjoy it because writing the grant gets the ideas down on paper and forces me to make a plan. And that's not a bad thing.
Katie, I know you had a question that I had really thought was a useful question to ask. Would you mind asking the question?
Yeah, of course. It kind of relates to the whole science on Twitter and mainstream media sort of thing. So my question is, how can science and research, especially within the field of neuroscience and mental health, be best served by mainstream media today? Because there seems to be a fine line between oversimplification and overgeneralization for and by the general public versus informing by presenting the complex facts. So what I'm getting at is the balance between benefits and risks associated with public person. And going off of that, from what you've seen, is there a neuroscience research area that is being represented?
Katie, you're cutting out a little bit. So I think we heard the gist of the question about communicating science to the public and this fine line that you walk between full disclosure and actually accurately describing the science versus communicating it in a simple enough manner? And then what was the last thing you said, Katie?
The last part of the question was, what you,
Oh, no.
is there a neuroscience research area that's actually being misrepresented?
Oh, okay. I don't think we heard that, but, oh, the internet is going bad. Okay, thanks, Rachel. Katie, you can type it in the chat, but, Dr. Heilbronner, did you hear that well enough to answer at least the first part of it?
Yeah, I can answer the first part. Oh my gosh, that's unbelievably difficult. I have not taken a major crack, honestly, at science communication. One of my grad students is very interested in that and takes coursework on science communication. I am contacted not irregularly by reporters to try to comment on a particular topic or something, and I really struggle with this. If anything I say feels like an oversimplification, and when it's what people often want is for us to provide advice, and that is where I get really, really nervous about what the brain findings mean for living your life, for education, for mental health, for things like that. What do they mean for you, the reader, reading right now this article? That's really tricky because often a lot of what we do doesn't really have value judgments. It's just, how does something happen, right? And so figuring out what would make someone happier is challenging. It often feels like if it's not oversimplified, and if you refuse to do that, then you just wind up telling fun little stories. And it's like, some of those are fun, but maybe not, maybe they're not interesting or even very, very relevant. Is that really what we want to communicate? This is hard. I wish I had an answer. Let me check the chat. Oh, is there a neuroscience research area that's being misrepresented in the mainstream media? I don't know about mainstream media, but there's some not so great neuroscience that's filtered into education. I know this, my mom was a teacher for many, many years, and then went back and got her PhD and is now in educational psychology. And she will sometimes text me in the middle of a presentation where somebody is trying to apply neuroscience research to education. There is legitimate research on that area out there, but a lot of what, I think there are some more, there are organizations that maybe whenever I see what they're promoting, it just doesn't seem right to me. It's sort of like left brain, right brain stuff. There's a lot of that type of stuff. Yeah, anything about left brain, right brain, unless they're specifically talking about language, you should not believe that. Like, okay, language is lateralized to the left side of your brain. There's been a lot of studies trying to figure out other things like that, but I would not, it's not like rational versus, emotion driven or whatever these, or creativity, I guess that's what it is. It's like logic versus creativity. That's not true. None of that is true. But I think a lot of mainstream media knows that and is more sophisticated than that by now. It's not so, when I see neuroscience coverage in the mainstream media, it's not usually that it's wrong. It's just, it's hard to convey the point, what it should mean for the listener or the reader, because it's often part of some really broad research program, which you guys have sort of been exposed to, that does eventually have a goal, but it's hard for it to be an immediate goal for the person that's ingesting. So yeah, I don't know. I wish I had an answer. I think science communication is its own field and belongs to its own, its own experts. And of course, a lot of that we've come up against during the pandemic. And I think a lot of attention is going toward that right now, rightfully so.
Can I ask what kind of process you undergo to like devise an experiment?
Oh, um, hmm. What do uh, it's different every time. It's not like I have, okay, today I'm going to sit around and think of an experiment, but the ideas often come up in conversation with other members of my lab or with other faculty or something like that. And then they eventually develop into something actionable. But that process takes a lot of meetings. So I would say science involves more talking than one might think. I mean, you could go it alone, I guess, and try to come up with these ideas just by reading the literature and on your own in the lab implementing them. But I think that a lot of people would miss things that way. So I guess my way of devising experiments and making plans involves talking, more social interaction than one might believe.
We have time for probably like another question or two.
I got a question as well.
Oh, okay,
Go ahead.
1, 2.
Yeah.
Okay, mine's just a general kind of like, what do you think the future of neuroscience looks like? Like what area or field do you think is gonna advance the most in the next coming years?
Oh, I hope it's that non-invasive neuromodulation for humans. That's, to me, that's a big part of the future. Some of those genetic techniques, so I should say like virally introduced genetic techniques are going to be applied to humans, I think. Gene therapy and things like that. I hope we see advances in neurodegenerative diseases as well. I think that neuroscience has also already moved toward much larger data sets. So if you're doing an electrophysiology project, you record from way more neurons at once. If you're doing a human neuroimaging project, you consolidate across multiple sites and look at way more subjects than we used to. It used to be that an MRI experiment had like 12 subjects in it, and now they often have hundreds to thousands. But they're not redoing those scans for every study. They're central repositories. So cross-institution, cross-site work is becoming incredibly common as well. I think everything is just getting bigger. So just like more data on that side.
So I was wondering, has COVID-19 had an effect on your ability to do research over the last year?
Yeah, it's been very challenging. So the anatomical work and the diffusion MRIs, those are things that can only happen in person. Like I can't do histology, which is how you stain the tissue to visualize the tract tracers, over Zoom, and I can't do it from home. And so let's see, Today is the day I locked down my lab, the 1-year anniversary, actually. So on March 12th, 2020, I told my lab we were all present. I had told them earlier in the week to only come in if they had something in person to do, if they needed to read or write, to stay at home, but they were still all in person because they all had in-person stuff to do. And late in the day, I said not to come back the next day. It was just a few days before the university decided to lock down, but Tom Hanks freaked me out, I guess. Do you guys remember when Tom Hanks, yeah, so that was, yesterday was that anniversary. So it was today when I told my lab to go home. I went home as well, and then we didn't come back for 2 months, except I went in to make sure the freezers hadn't died because we had tissue. So my histologist and I, my lab manager and I, we were classified as essential employees and were able to just go in and check the freezer and nothing else. I also donated all of our masks, gloves, syringes, gowns, surgical gowns, to the COVID effort. So they emailed us and asked if we had anything, and we just set donations. I set a whole room full of donations outside the door. I don't know where they went, but they're gone. So after those 2 months, the university implemented like a graded sunrise plan. So I was allowed to have people back in the lab at 50% capacity. The problem is that like the surgical procedures that we do and the MRI procedures require you to be close together, less than 6 feet apart. And so we were still uncomfortable doing that. In addition, an interesting feature, while we could get masks, we could not get surgical gowns, since I had given all of ours away at the beginning when they were needed. All the factories that produce surgical gowns started making masks. And so we couldn't get them for any price for several months. So we just couldn't do big chunks of our procedures. So we were able to do a little bit of histology and a little bit of microscopy, but nothing that required more face-to-face interactions. And those rules have gradually loosened and then tightened again over time. Yeah, it put us back a lot. My grad students, I know it's been challenging for them. They had a whole course of experiments planned and they were able to do some tiny fraction of that. So we've for them, there's one that, we've made different plans depending on what their personal goals are. If they feel like they need to finish sooner, we changed their experimental plan to be working more with existing data. If they wanted to stick around longer, we just sort of expanded their time horizon. Like we're working with them to make that happen. It's been, it's been incredibly disruptive to research in general, just across the world, across the nation. So my lab is not unique in that respect. There have been quite a few studies showing that the impact has been greater on people with wet labs, meaning labs where you have to like physically do work in person versus dry labs where you're like working with data that already exists or something like that. The impact has been greater on people like me who are relatively new faculty versus much more established faculty who maybe could just write papers that were like collecting dust on their desk or something like that. The impact has been greater on women than on men. The impact has been greater on caregivers than on non-caregivers. So, there are a number of studies that validate everything that we saw in numbers, but that doesn't mean that we fixed it. So I'm sure that you guys are also feeling the impact in your own way. And so, everybody will, at the end of this, have their own pandemic stories, and we'll all have to reckon with those on the other side. We should end on a happier note. Yay!
Yeah, I was just contemplating.
Yeah, no, so hopefully some of you are planning to continue in neuroscience. Yeah, are any of you, I guess I heard one person is going to graduate school. People have been putting together resources. I've noticed there are more of these now. If you feel like you need to get more research experience before you apply to graduate school, that's a typical route. Hopefully you've sort of heard about that, to be a research technician for a few years in somebody's lab. I'm going to put in the chat. There are a couple, you know what, I'm going to email Dr. Schaefer because I'm not going to be able to find it fast enough. There are a couple of resources for finding those now, because those aren't always like having to look at every university's website might just be right? Like that doesn't really work. So the people have gathered these for the field of neuroscience, and they're sort of posted in a few places. So I would encourage you to take a look at those if you feel like you want to continue, if you feel like you think you might like to do research in an academic setting, but you're not sure. For those of you that are younger, I don't know whether you've heard of, we do run a neuroscience summer program it's the Life Sciences undergraduate research program at UMN. And so I would encourage you to take a look at that.
Wonderful. Michaela, I'm going to give you one stab at a question. It looked like you were really trying hard to hit your mute button there.
Yeah. Okay. My one final question for you is like, what is one like goal you have for your career or something you hope to accomplish before your career ends?
I have one goal for the whole career, which is that I want to create the wiring diagram of the human brain. And I will use any any hack to do that, I will, I showed you the pipeline that I'm doing right now, which is like to use diffusion tractography and try to correct it. And so that'll get me like part of the way there. It's not going to get me all the way there. And so, I have my eye on technologies. There's nothing to help me right now, but I will just keep hacking away at that pipeline in order to, I want to like be able to visualize all of the connections of the brain. I'm not alone in this, but that's the prize. I want to keep my eye on the prize.
I love that. What a great end to this discussion. Thank you very much, Dr. Heilbronner, for
having me.
It has been
wonderful, amazing questions. You guys are so engaged. I'm very impressed.
So thank you. Have a nice Friday and have a nice weekend and enjoy being back in the lab.
Yeah.
Finally. Yeah. All this time. Yes.
Thanks.
Thank you.
Thank you.
So students, good work. Great questions. I'm gonna stop the recording.